Hello, my name is Juliann Savatt. I'm a genetic counselor at Geisinger, and I help to coordinate Clin's patient data sharing efforts. Thank you all for attending this breakout session today. I am delighted to speak to you about GenomeConnect, the ClinGen patient registry, and our patient registry partnerships. I do not have any potential conflicts of interest to disclose. Before I jump in today, I want to give you a sense of what we'll be talking about during today's session. I first hope to explore how patients can share their genetic and health information through patient registries, including GenomeConnect, and the other registries that we're working with through the ClinGen patient data sharing program. In addition to discussing how patients can share data, I want to explore how patient-centered data sharing can benefit the genomics community and individual patients. We'll describe how patient-shared data can support and aid in variant classification and gene curation efforts, and we'll describe how individual patients can benefit, including through the ability to partake in external research, the ability to receive updates about their genetic results, and the option to connect with other stakeholders, including other patients and families. I know you've seen this graphic a few times over the past few days, but I want to give you a sense how these efforts fit into the broader ClinGen work. As you know, the Clinical Genome Resource, or ClinGen, is dedicated to building an authoritative resource that defines the clinical relevance of genes and variants for use in precision medicine and research. Ultimately, the goal is to understand what genes and variants are associated with disease and how genomic information can inform clinical care. These efforts to answer these questions rely on data sharing. ClinGen encourages data sharing from a number of stakeholders, including patients, clinicians, labs, and researchers. And today we'll be focusing in on efforts within ClinGen that engage patients in data sharing. Laboratories, researchers, and clinicians have been sharing genomic and health data for some time, but ClinGen felt strongly that patients could also serve as an important source of additional information. Patients are often experts about their own health history and can provide much more detail about their symptoms over time, their diagnoses, and an overall picture of their health. ClinGen felt that access to this additional health information could help increase our understanding of genes and variants. As a result, in 2014, the ClinGen team launched a patient registry called GenomeConnect to enable patients to share their de-identified genetic and health information with publicly available databases. GenomeConnect is open to anyone who has had genetic testing, regardless of results or diagnosis, so it is not gene or disease specific. Participants enroll online, consent to de-identified data sharing and Recontact, provide their health history through an initial health survey and additional surveys that might be assigned over time, and upload a copy of their genetic test results so that our team can pull out the relevant variant information. To give a bit more information about how health history is collected, as I mentioned, after consent, each GenomeConnect participant receives an initial health survey that's used to capture an overview of their health. The initial survey inquires about general issues across 17 broad body systems. It's really like a review of systems that might be completed in a doctor's office when you go for a visit. For example, the survey asks, do you have any issues with the nervous system, the digestive system, the respiratory system? Each question asking about a broad system includes examples of issues in that system using patient-friendly language. Then, after those initial questions, more specific questions are assigned based on any systems or categories that the participant indicates they've had an issue with. We also assign additional surveys over time to capture even more detailed information. For example, if a participant indicates that they have a history of issues with the cardiovascular system, they'll receive a more specific question on that initial survey asking for specific issues with the cardiovascular system. If they select a history of cardiomyopathy, we'll assign an additional survey down the line that asks them more information about the type of cardiomyopathy, any additional risk factors for cardiomyopathy, and any treatments they've had. Although the survey is in patient-friendly language, on the back end, participant responses are automatically mapped to Human Phenotype Ontology terms, or HPO terms. These terms are structured to enable data sharing outside of the registry. For example, if a GenomeConnect participant indicates that they have had growth differences and goes on to select short stature, you can see its patient-friendly description there on the screen, On the back end, it would map to this HPO term, again, enabling for structured data sharing. Participants share their genetic test results by uploading a PDF or image of their result, or by providing GenomeConnect with permission to request their records from the lab that completed their testing or their healthcare provider. Genetic counselors on our team review reports once we have them in hand to ensure that all of the genomic data are collected in a standardized way and to ensure its accuracy. The information we collect includes things like the genome build, the gene and transcript, the variant classification from the reporting lab, and the variant details, including the CDOT and the PDOT. Once genomic and health information are collected from patients in the registry, their de-identified information is shared with Clin, a publicly available database of genetic changes and their classifications. This database is where many clinical laboratories submit variants. GenomeConnect submitting information to Clin makes this information publicly accessible to other researchers, labs, and healthcare providers. The submission from GenomeConnect is done in such a way that it's clear that this submission is coming from a patient registry, that the health details are participant reported, and that the variant information and classification that we're providing are as they are on the participant's report. We are not attempting to reclassify or reinterpret that variant in our submission. Additionally, a subset of participants share reports that have identified a candidate gene. A candidate gene is a gene that does not currently have a clear disease association, but based on preliminary research of the gene's role in the body or a small number of patients, there is some suspicion that it might be associated with Mendelian disease. More research is needed, however. Whenever candidate genes are shared by participants, these are also submitted to a data network called Matchmaker Exchange to try to enable matching with other healthcare providers, labs, and researchers that are exploring gene-disease relationships. Our hope is to move those candidate genes, or help support moving those candidate genes, to more definitive categories of association with Mendelian disease or no association with Mendelian disease. So why is patient-centered data sharing important? GenomeConnect affords patients in the genomics community three main benefits that I want to walk through today with some case examples. The first benefit, patient-centered data sharing can inform variant classification and gene disease validity. As you know, when a genetic testing lab, researcher, or clinician has a variant in hand, the next step is to try and understand if that variant is associated with disease. The first question is this particular gene associated with disease? Do we have enough information to say that this gene is associated with this Mendelian disorder? If not, it means that more information is needed, more cases of variants in that gene, more assessment of that particular gene. If we do know that a particular gene is associated with a Mendelian disorder, the next question then is this variant pathogenic? Do we have enough information to say that this particular variant would impact how the gene works in the body? Answering these questions relies on pooling many sources of information, population databases, the literature, information in Clin to try and make these determinations. Information that's shared by patients can be a part of the data that are pooled to try and answer those questions of, is this gene associated with disease? Is this variant pathogenic? Patient data sharing can provide, first, access to additional cases that could be used to inform a gene-disease relationship. Additionally, patients can provide case-level details, information about their history that could be used to inform a variant classification. Participants in GenomeConnect can provide additional health information above what's typically shared by laboratories or clinicians who are submitting to Clin. We also provide information regarding inheritance of the variant when available, and we have access to information about other variants that were reported through that participant's testing. Finally, we have the ability to recontact patients over time if additional information would be needed. Knowing patients' specific symptoms, ages of onset, inheritance of the variant, what other variants were found, all of this information can help us better understand variants and their relationship to disease. In addition to providing those additional details like enhanced phenotypic data, information regarding inheritance, patients enrolling in GenomeConnect also are providing novel genomic information that has not been submitted to Clin previously. From our submissions to date, 41% of genetic changes were not submitted by the reporting lab or another submitter, meaning GenomeConnect was the first time that this variant was put out there in the database. GenomeConnect sharing information in the public space can be useful the next time that genetic variant is identified, or if similar variants are found in other patients. Ultimately, the information from GenomeConnect can be used to understand gene-disease relationships and variants' pathogenicity. To illustrate that patient data sharing can provide valuable data to increase our understanding of genomics, I wanted to share a case example from our experience with GenomeConnect. For the purposes of our discussion today, I'm going to call this participant Ella. Ella is a pediatric patient with a history of seizures beginning in infancy. She had exome sequencing ordered back in 2015, and a likely pathogenic variant in SCN8A, a gene associated with infantile epileptic encephalopathy, a seizure condition, was found. Her parents were tested as part of that exome, and the variant was not found in either of them, suggesting that the change is de novo or new and unique to Ella. Because the genetic change hasn't been seen in healthy individuals, was found to be de novo, and was expected to impact the protein, the clinical lab, Lab 1, classified it as likely pathogenic. Ella and her family had a likely explanation for her history, but they chose to participate in GenomeConnect to get more information out there in the public space. We submitted Ella's de-identified genetic and health information to Clin. Our submission would have included the laboratory that reported the variant, Lab 1, their classification, likely pathogenic, their report date, the fact that the variant was de novo, and health details that Ella's parents had provided. At the time of our submission, there were two other records in Clin. The first was a record from Lab 1, the lab that had reported the variant out to Ella and her family. Although Lab 1 has clinical details about Ella and is aware of the variant's de novo status, these are details that weren't included in their Clin submission. Laboratories often have these details, but often are unable to share them with Clin. A second lab, Lab 2, also had submitted the variant to Clin, but based on their information and what they had available, they had classified the variant as a variant of uncertain significance. After our submission, Lab 2 reached out to our team to ask more about Ella and confirm the variant was de novo. In their patient, they were unable to perform parental testing to get that sort of information. As you know, understanding if a genetic change is de novo can be incredibly helpful in informing variant classification. We were able to confirm by re-reviewing Ella's report that the variant was in fact de novo, and were able to share this back with Lab 2. Lab 2 then reclassified their variant as likely pathogenic and updated the patient's report. Ella's results in care were not impacted by her family taking the time to share her results, but ultimately them sharing enabled another family to get clear genetic test results and to inform variant classification for them. This is just one case, but I think it illustrates that patient data sharing can be incredibly valuable in increasing our understanding of genes and genetic variants. In addition to informing variant classification and gene-disease relationships, patient-centered data sharing can enable individual participants to receive genetic updates. As I just outlined in that case example, our understanding of genetic changes can shift over time. In fact, we hope to reclassify uncertain variants as benign or pathogenic as more information becomes available. And we know from our practice that the understanding of pathogenic or benign variants might also shift as new information becomes available. One study of a single cancer genetic testing laboratory here in the United States found that over a 10-year period, 6.4% of variants that they had reported were reclassified at least once, leading to almost 60,000 reports being updated in that time. We know variant reclassifications occur. We're continuing to learn in genetics, and as a result, our understanding can shift. Clinical laboratories and healthcare providers' duty to reassess genomic results and recontact patients has been the subject of ongoing discussion. Here in the States, although there's no clear legal duty to recontact, there is a consensus that recontact is beneficial and that most patients value being recontacted when there's updated information about their genetic results. Laboratory and healthcare providers' reassessment of variants and their recontacting practices vary, and there are certainly barriers to recontact, including limited time and resources. Under current circumstances, two large genetic societies, the American College of Medical Genetics and Genomics and the European Society of Human Genetics, have suggested engaging the patients in the responsibility of recontact as a means to address potential barriers and to help ensure patients are receiving up-to-date information. Early on in GenomeConnect's data sharing, we recognized that the registry might identify cases where the participant's report is out of date compared to the current knowledge base, and that GenomeConnect could provide a means for patients to engage in the recontacting process. For instance, through our data sharing with Clin, the GenomeConnect team might identify that the patient's genetic test report and the classification included is out of date compared to the clinical testing lab's current classification or an expert panel's classification. Through our submission to Matchmaker Exchange, we might learn that a gene-disease relationship has been clarified and that a gene that was once a candidate gene now is associated with a Mendelian disorder. This information may not always have made its way back to the patient or family that has enrolled in the registry. Recognizing that GenomeConnect could identify updates to participants' results through our data sharing, we started to explore if we might be able to share such information back to participants. In September of 2016, we sent a survey to current participants in an attempt to capture their preferences regarding receiving updates about their genetic test results in general and receiving such information from the GenomeConnect registry. Our survey was sent to the participants enrolled in the registry at the time, and we had a response rate of approximately 20%. Participants were provided with a brief summary about genetic test results and how results might be updated over time. They were then asked up to 8 questions regarding whether they would like to learn about these updates and if they would like to learn about these updates from GenomeConnect. Of those that completed the survey, 99% indicated that they would want to receive updates in general and that they would want to receive them from GenomeConnect if we learned of such information. In fact, 98% of participants in the survey saw potentially receiving updates regarding their or their child's results as a benefit of participation. The 2 participants that were unsure if they would like to receive such updates from GenomeConnect were asked why, and responses included that they would prefer to receive updates from their healthcare provider, or they felt that the registry would not be able to provide enough information about updates that were identified. Also, they were unsure or not interested in receiving updates in general. In thinking about providing updates back to participants, we realized that some updates may impact medical management, while others may not. For example, a change from VUS to pathogenic might impact someone's diagnosis and clinical care, while an update from likely pathogenic to pathogenic is unlikely to impact their management. Consequently, we asked participants what type of updates they would like to receive. The vast majority, 92%, indicated that they would want any and all updates. In addition to obtaining information on preferences about the type of updates that are provided, we inquired if receiving updates should be something participants have the option to decline. The majority, 82%, indicated that there should be the option to decline receiving such information. So while they wanted updates from the registry, they recognized that choice upon enrollment was important to them. Following this survey, GenomeConnect created a process to provide participants the option to receive information regarding updates to their variant classification or gene disease validity. In February 2017, GenomeConnect established a process that was really informed by those survey results. Upon enrollment, individuals are now required to provide their preference regarding receipt of updates from the registry. Participants that enrolled prior to February 2017 were prompted to update their preferences via email, and reminders have been included in the registry's quarterly newsletters. To date, 98% of those who've responded have opted to receive updates from the registry. If an update is identified, the reporting lab is first contacted to confirm that the classification in Clin or the gene-disease relationship identified via Matchmaker Exchange do reflect their current understanding and classification. This also enables report updating if needed. If the participant has opted into updates, they are then informed via email that there might be an update to their genetic test results. The email directs them back to their healthcare provider or a genetics provider in their area and provides information that would be helpful to share with their healthcare provider. Finally, the email reminds them to upload any updated genetic test reports so that the registry can update our database in our ClinVar submission. This brings me to my second case of a GenomeConnect participant that I want to dive a bit further into. I'll call her Katie. Katie is an adult with a history of seizures and an intellectual disability. Katie's mother has legal guardianship and chose to enroll Katie in GenomeConnect after she had had several genetic tests, including exome sequencing. Katie's exome sequence and all of her other tests were not diagnostic, though exome did identify a variant in a candidate gene, CSN-K2B. As we discussed earlier, candidate genes are those that do not yet have a clear association with the Mendelian phenotype, but based on preliminary evidence, are candidates. Because a candidate gene was identified, after this family enrolled in GenomeConnect, that gene was submitted to Matchmaker Exchange and the variant to ClinVar to get it out there into the public space. In 2019, through our Matchmaker Exchange submission, GenomeConnect became aware of a relationship between CSN-K2B and mild to profound intellectual disability, developmental delays, and various types of seizures. We contacted the reporting lab, who shared that they had upgraded the CSN-K2B gene to disease-associated, and recently reported the same variant out as pathogenic in another patient, but they hadn't shared updated information back with this particular family. We then contacted Katie's family to let them know that there might be an update. They circled back with their healthcare providers and received an updated report from the laboratory, which ended this family's diagnostic odyssey and provided an answer for Katie's history. Another example where GenomeConnect facilitated updates is case 3, who I will call David. David is a pediatric patient with a history of a prenatal cystic hygroma who now has behavioral issues, fine motor delay, and dysmorphic features reported by his parents. David underwent exome sequencing in 2015, and no causative variants were identified. 2 variants in DHCR7 were found. 1 variant was found on each copy of the gene. 1 was interpreted as pathogenic, and the other as uncertain. As many of you may know, DHCR7 is a gene associated with an autosomal recessive condition called Smith-Lemli-Opitz syndrome. Smith-Lemli-Opitz syndrome is characterized by distinctive facial features, small head size, intellectual disability or learning problems, and behavioral issues. After David's family enrolled in the registry, we submitted these variants to ClinVar, and through that submission, learned that the variant of uncertain significance had been reclassified to likely benign by the reporting lab. The family had opted to receive updates, so we recontacted them, telling them that there might be updated information, and this prompted them to speak to their ordering provider. Their ordering provider then ordered an exome reanalysis, allowing the family to receive an updated report documenting this variant reclassification, but also identification of a novel de novo variant of uncertain significance in another gene associated with dysmorphic features, neurodevelopmental disorders, and congenital anomalies. Recontact through GenomeConnect can provide answers to some families, like in Katie's case. But even in cases where the update provided by GenomeConnect does not provide the family with an explanation, like in David's case, it still can be important. In this case, Recontact prompted additional assessments and led to the identification of another variant of interest. Updates such as this reclassification from VUS to likely benign are not often prioritized by labs under current constraints, but as this case illustrates, can be important to individual families. In total, GenomeConnect has provided 47 updates back to interested participants to date. They are highlighted here on this slide. Of those 47, nearly a third are likely to impact a participant's diagnosis and/or care, though the remaining still might impact patients' care, like in David's case. All in all, patient data sharing can enable individuals and families to remain informed about their genetic test results. So finally, patient data sharing can support connections between stakeholders. GenomeConnect enables connections with other researchers or clinicians interested in a particular gene or variant. If a researcher interested in a gene reaches out to GenomeConnect, we are able to recontact the participant to collect additional information about their health history or to connect them with additional external research opportunities. Additionally, participants can match with one another through the registry based on factors like gene or diagnosis. Many genetic conditions are rare, and patients might have the experience of being told that they are the first patient their healthcare provider has seen with that diagnosis, or the first one with a variant in the gene that they've cared for. The registry can enable them to connect with others with similar results or diagnoses to enable the exchange of both informational and emotional supports. To highlight how GenomeConnect can connect patients with other stakeholders, including researchers, I wanted to share an additional case from GenomeConnect. In March 2016, a patient who I will call Daniel underwent exome sequencing due to a history of an autism spectrum disorder, intellectual disability, Tourette syndrome, and hypotonia. No diagnosis was identified, but 2 candidate genes were found. In 2019, the family enrolled in GenomeConnect, and the candidate genes reported were submitted to Matchmaker Exchange. In June of last year, a researcher in Europe contacted GenomeConnect because they saw our Matchmaker Exchange submission and were working to put together a case series reporting on patients with variants in GRIA1, one of the variants identified in this patient. We were able to recontact the family to let them know about this case series and, with their permission, connect them with the researcher for inclusion in the publication. Through GenomeConnect, this registry participant was able to connect with a researcher interested in one of the genes identified through their testing and contribute to our understanding of this particular gene. So I hope I've given you a sense of the power of patient data sharing and the benefits to the broader genomics community and individual patients. Because of these benefits that we've seen through GenomeConnect, we're working to expand opportunities for patients to share data by working with other registry partners. We're working with external registries through what we call our Clin patient data sharing program. As you may know, GenomeConnect is just one of many patient registries. There are many other gene and condition-specific registries out there who have been collecting valuable genetic and health information, but it might not always be shared publicly. It might sit within the registry and not be as useful as it could be. We are now working with registries who are engaging patients who've had genetic testing to help offer them the opportunity to share their data with Clin and other databases. GenomeConnect works with registries built on the same platform as GenomeConnect, as well as registries that are built on other external platforms. For those on the same platform as GenomeConnect through Invitae, we're able to use an established process to enable data sharing. Essentially, we're able to copy over things like pieces of our consent, how we curate genetic test reports to enable data sharing for those registries. For registries on other platforms, we take a case-by-case approach to determine how we could best support data sharing, to explore how do we obtain consent from those participants, how do we collect genetic information, and ultimately how it's shared. In the end, we're working to enable de-identified sharing of genetic and health data with ClinVar and other databases, and we work, when possible, to offer some of the same benefits back to registry participants, like the ability to receive updates about their genetic test results. To date, we've engaged with 10 registry partners to share data. The registries are depicted on this slide, and as you can see, some are gene-specific registries, like the Grace Science Foundation's NGLY1 Deficiency Syndrome Registry. Others are condition-specific, such as No Stomach for Cancer, that's engaging patients with stomach cancer, or Simon Searchlight, that's engaging patients who have autism spectrum disorders. Finally, some registries are not gene or condition specific, like the Invitae patient registry that engages those who've had genetic testing. Altogether, GenomeConnect and these partnerships with external registries have enabled us to engage over 4,600 patients in data sharing and have resulted in the submission of over 1,900 unique variants to ClinVar. This graph here depicts our enrollment over time. So you can see the blue line is GenomeConnect, where we started to engage participants in October of 2014. And then the data sharing program is the green line, and we started engaging with external registries in mid-2018 after we had established our data sharing practices within GenomeConnect. Although ClinGen is supported by an NIH grant, ClinGen is not just working to engage patients within the United States. We want to support data sharing from around the world. As I alluded to earlier in my presentation, sometimes we're restricted by local data sharing regulations, but in the vast majority of instances, we're able to accept participants from around the globe. Currently, 10% of our participants are from outside the United States, and participants enrolled in data sharing are from 52 countries, including a small number of participants who indicate they reside in a country in Africa. So how can you get involved in patient data sharing? First, you can use the information shared by patients to inform your understanding of the relationship between genes, variants, and health. Remember, variants and health details from participants are periodically shared with ClinVar, and candidate genes are submitted to Matchmaker Exchange. You can use those resources to explore what data we've submitted, and if you have questions about additional details from a submission or aren't sure if you might have missed a variant in your gene of interest, you're welcome to contact us. We're happy to engage with clinicians, labs, or researchers and let them know if we have details that might be of use to them. First, use the information shared by patients to inform your assessments of gene-disease relationships and your variant classifications. Remember, variant and health details are periodically shared with ClinVar from GenomeConnect and the registries that we work with. Use ClinVar to explore what information has been shared to date, and you're always welcome to recontact us if you have additional questions about a submission, if you're interested in recontacting a participant for additional details, or if you are interested in knowing if we've seen a particular variant and might not have submitted it to ClinVar just yet. We welcome contact from clinicians, laboratories, and researchers. To help illustrate what details are shared by GenomeConnect and where to find them in a ClinVar record, I thought we could walk through one example on the call today, just briefly. So once you search a variant in ClinVar, you're taken to a page such as this. If you scroll down, you can see the individuals or institutions who have submitted the variant. In this case, GenomeConnect is one of the submitters, the fourth one listed here. Because GenomeConnect does not classify the variant as part of our data collection or submission to ClinVar, there's no interpretation listed here on the main page from the registry, though we do provide the classification from the reporting lab, the reporting lab's name, and their report date. On this main page, you can see some other details, including inheritance of the variant, if available, from the participant's report, and some additional details in the comment regarding who reported the variant and how did they classify it. Here you can see that Genetic Testing Registry ID 26957 reported the variant out as likely pathogenic in June of 2018. You can see additional information if you click on Evidence Details, which will take you to a page that looks like this. The specific columns might look different on your particular display, but you always can edit them by hitting Choose Columns here. You can see additional items on this page, including clinical features in the individual who this variant was identified in, their age range, their sex, and then structured details again about that reporting lab, their classification, and their report date. You're also, as I said, always welcome to email our team with questions about a particular variant or to recontact participants over time. If you're interested in browsing all the submissions from GenomeConnect or the registries that we work with, you can go to the ClinVar submitter list at the URL on the slide. All submissions from GenomeConnect or the registries that we work with have GenomeConnect in the name. GenomeConnect is named GenomeConnect ClinGen, and then the registries we work with have both GenomeConnect and that particular registry's name. So they're easy to find on this page if you just look for GenomeConnect. Also, if you work with patients as a healthcare provider, researcher, or in a testing laboratory, remember that you can help work with ClinGen to expand data sharing. If you work with individual patients, inform them of opportunities to share data through GenomeConnect. As I mentioned, we want to engage as many patients as possible in these efforts, and patients who are engaging might benefit as well with updates or the ability to connect with others. If you work with a patient registry, discuss the opportunity to engage in the patient data sharing program. We would love to be able to discuss with them how we might be able to help support submissions to Clin and other databases. In summary, I hope you've come to understand that GenomeConnect and other patient registries offer a structured way for patients to share their genetic and health information. Sharing that information can help inform our understanding of gene-disease relationships and variant pathogenicity. For individual patients, sharing data can also help them remain informed about their genetic test results, can let them know about additional research opportunities, and can enable them to connect with other patients and families. Finally, Clin is actively working to expand data sharing efforts to offer more patients the ability to share data. In your work, you can support patient data sharing by using the details that are shared by patients to inform your gene-disease relationships assessments and your variant pathogenicity. Also, you can encourage patients to share data by enrolling in GenomeConnect or by working with our team to explore if we're able to help support data sharing from a patient registry that you work with. With that, I want to acknowledge all of the people that helped to make GenomeConnect and the Clin patient data sharing program possible, including our 4,600 participants and the broader team at Geisinger and the Broad. You can find more information about these efforts on the Clin website at the URL on the slide. You can get there with the QR code as well. Additionally, we've attached some resources to today's meeting that might be helpful. And if we're not able to answer all of the questions during today's discussion, or if you have additional questions after today's presentation, you can contact us at info@genomeconnect.org. So with that, thank you, and I think we can turn it over to questions.